Okay. Okay, so today we're going to have our last problem set. Question 1 and 2, we're just going to be doing 2 questions for PS4. Okay, so question number 1. So, Dr. X sees a patient who presents with hypoglycemia in long periods, 20 hours, between meals. Dr. X orders a test which shows that there's an accumulation of fructose 1,6-bisphosphate. A metabolic panel showed fructose 1,6-bisphosphate is found to be at normal physiological concentrations, but functioning at lower activity levels. Moreover, downstream gluconeogenic enzymes such as glucose 6-phosphatase displayed negligible activity. Pyruvate carboxylase and phosphoenolpyruvate carboxykinase have normal activities in liver biopsies. In the periods between meals, Dr. X's supervisor, Dr. Y, reminds her that glucagon stimulates glycogen degradation. So Dr. X administers glucagon to the patient, who was well fed prior to the test and finds that blood sugar rises in response as normal. Dr. X prepares a report for Dr. Y in which she identifies which pathway and which specific enzyme is defective in the patient. What are Dr. X's conclusions, and explain your reasoning? So we know that the glucagon pathway, it starts the gluconeogenesis pathway, which is the reverse of the glycolytic cycle. And these enzymes, pyruvate carboxylase and phosphoenolpyruvate carboxykinase, are, and this one, fructose 1,6-bisphosphatase, are part of the gluconeogenesis cycle. That's why it says gluconeogenic enzymes here. And this one too, glucose 6-phosphatase. Okay, I'm going to move on. So this was the question, problem 1. And we have to answer this right here. Okay, so the problem states that pyruvate carboxylase, phosphoenolpyruvate carboxykinase, and glycogen degradation are functioning normally. So we know that hypoglycemia cannot be attributed to any of these pathways because fructose 1,6-bisphosphatase is found to be functioning normally, but at lower activity levels, we can assume that it is being inhibited by something. In the reciprocal regulation mechanism of gluconeogenesis and glycolysis, we know that fructose 2,6-bisphosphate inhibits fructose 1,6-bisphosphatase. Therefore, we can infer that fructose 2,6-bisphosphatase is deficient. So basically, if you guys remember in the last problem set, how I said when we had the glycolytic pathway, like if there's, for example, glucose here and then hexokinase here, an enzyme, then hexokinase will inhibit the glucose product. Does that make sense a little? Okay. I'm going to explain it more on the next slide. So the explanation, this one's a little detailed, but I'll go ahead and read it. Okay, so after eating meals, excess fructose 6-phosphate is converted into F2,6-bisphosphate by phosphofructokinase 2 to upregulate glycolysis and inhibit gluconeogenesis. During low blood sugar levels, phosphofructokinase 2 is phosphorylated into fructose 2,6-bisphosphatase, which converts fructose 2,6-bisphosphate into fructose 6-phosphate. Dysfunctional fructose 2,6-bisphosphatase causes an accumulation of fructose 2,6-bisphosphate, which, as mentioned before, inhibits fructose 1,6-bisphosphatase. So in order to understand this, you kind of have to know the glycolytic and the gluconeogenesis cycle. Specifically this one because it has enzymes specific to just the gluconeogenesis cycle. And just remember that these two, glycolysis and gluconeogenesis, are antagonists of each other. So glycolysis is the forward reaction from glucose to pyruvate, and then gluconeogenesis is the reverse reaction of glycolysis, which is going to be essentially glucagon, and then it leads to glucose. Or like pyruvate to glucose. But it is an agonist of glucagon. Does that make sense? Okay. I'm going to move on. So keep in mind, glycogen degradation can raise glucose levels, but after the 20-hour mark, glycogen stores are depleted and gluconeogenesis is used. Pyruvate carboxylase and phosphoenolpyruvate carboxykinase are functioning normally, so gluconeogenesis is able to start but unable to compete. Okay. Another thing to keep in mind is that fructose 2,6-bisphosphatase is not missing. Otherwise, phosphofructokinase 2 would also be missing in the patient. Instead, a point mutation in the patient's DNA could have caused an inability to phosphorylate phosphofructokinase 2 into fructose 2,6-bisphosphatase. So this is just like knowing the gluconeogenesis mechanism and stuff. Okay. Does anyone want me to explain that better? Or I will move on. Okay. I'm going to move on. Okay, so this is some pictures for our question 1. So this shows the gluconeogenesis cycle. So it goes from fructose 2,6-bisphosphate, it's just like reverse. It's using this enzyme, and then the glycolytic cycle uses the phosphofructokinase 1 enzyme. And usually the kinase enzymes in the glycolytic cycle, this is just like a little key to remember, are irreversible. So that means they can't be used in gluconeogenesis. So glycolytic is an irreversible pathway and it is spontaneous. So the free energy is going to be way less than 0. It's going to be a large negative number. And then this just kind of shows the gluconeogenesis pathway because it has to do with glucagon, fructose 1,6-bisphosphatase, and then the phosphofructokinase, how they work together. Okay. But overall, for the diagrams, I feel like I would advise you guys to kind of ignore this and just stick to the answer, the paragraph answer that I gave you, and just like look at the cycle in general for the glycolysis pathway and the gluconeogenesis pathway, because this is a little confusing, but definitely still look over this a little. It's just like a little hard to see in relation to this problem. Okay, I'm going to move on. Okay, question number 2. This is the last question we're going to do for the day. So aspartate is an essential amino acid that helps in nervous system function as well as hormone production and release. It is derived from precursor oxaloacetate combined with glutamate. Glutamate production utilizes the reverse reaction of glutamate dehydrogenase. Glutamate production requires that an organism grows in the presence of oxygen. So here's our little diagram. Shows how we get to aspartate. And it's related to this problem. This is kind of part of the citric acid cycle, which you're going to see in a bit. Okay. So draw the simplest pathway from glucose to aspartate. So Dr. Spiro, like when he says simplest, he wants like the primary pathway. So in this case, it's like we know that it's going to go from glucose, it's going to go through the glycolytic pathway, acetyl-CoA, and then acetyl-CoA, as we know, enters into the citric acid cycle, or also known as the Krebs cycle. And we can see here oxaloacetate, and then it converts to aspartate. So you just draw glucose glycolytic cycle, and then acetyl-CoA, you draw the citric acid cycle, oxaloacetate, and just show aspartate. Okay, I will move on to question B if no one has any questions. Okay, what is the purpose of aspartate in the malate aspartate shuttle? So I would recommend you guys to know like the shuttles and stuff for this test. So here the aspartate converts back into oxaloacetate in order to transport NADHs in and out of the mitochondria as needed. This transportation of NADH occurs by cytosolic oxaloacetate converting itself into malate so that it can pass through the inner mitochondrial membrane and back into the oxaloacetate when it reaches the mitochondrial matrix. This makes oxaloacetate capable of producing NADH. So since the oxaloacetate cannot go in the inner mitochondrial membrane, as we said here, has to convert into the malate, right? And then it can go out into the mitochondrial matrix in order to produce NADH. So kind of just like dump it down or like use baby steps and like simplify it a little because these shuttles and transport systems are a little like specific. So just use like what's the purpose of a malate aspartate shuttle and aspartate. So what it does is aspartate is usually at the end product, it's the end product, and then it's a reverse reaction that leads back into oxaloacetate, and then it goes through malate, and then malate converts back to oxaloacetate to produce NADH. And that's how you can control how much is going to go in and out of the mitochondria. Do you guys want me to explain that more, or is that a little? I know this question was a little wordy, the answer to it. Okay. If no one has any questions for this, I'm going to move on to C. But I would advise you guys just to like look at it and at home, the shuttle a little more, like maybe watch a YouTube video and then you'll like get it a little more. But I think just like use like simple steps. Don't overthink these transport systems. Okay, so the last question, part 2C, aspartate production also requires a citrate synthase catalyzed conversion of oxaloacetate and acetyl-CoA to citrate. Include this reaction on your diagram and explain why it is necessary. So citrate synthesis is required in the production of aspartate as its downstream metabolite. Malate is a precursor to oxaloacetate in the citric acid cycle when coupled to the reduction of NAD+. Oxaloacetate is then transformed to aspartate via aspartate aminotransferase. With citrate synthesis preceding the citric acid cycle, intermediate alpha-ketoglutarate is also formed, which is a precursor for glutamate, which is another precursor needed for aspartate synthesis. This explained the necessity of citrate synthase in aspartate production. I'll wait a little and then I'll move on to the next slide and show you the picture so it makes more sense. Okay, I can move on. Okay, so this essentially we have to look at this. This is a simplified reaction, so it's just acetyl-CoA from the citric acid cycle plus oxaloacetate, which leads to citrate. And so overall, we have to see citrate synthesis is here. The citrate is here. And it's required in the production of aspartate, so because it leads to citric acid cycle. And we know that the oxaloacetate, in the diagram that we just saw, the oxaloacetate yields to aspartate synthesis, correct? So we know that its downstream metabolite, I don't know if I'm saying that right, but is going to be malate because malate is used to convert to oxaloacetate in the forward reaction, which converts to aspartate. And so, when it's coupled to the NAD plus and it converts to NADH, the malate here. It converts to oxaloacetate, correct? So then through this, the NAD plus and NADH, we need this in order to convert to oxaloacetate, and thus we can convert to aspartate. Via the enzyme aspartate aminotransferase. And then also, I showed you guys in the slides, let's see, here, so oxaloacetate and glutamate convert to aspartate and alpha-ketoglutarate. So that connects to how during the citric acid cycle, citrate leads to alpha-ketoglutarate, which yields glutamate here. And so that's going to lead to, it's a precursor that leads to oxaloacetate, which converts into aspartate. And then I think that's it. So citrate synthesis is required because it starts the citric acid cycle, and it leads to alpha-ketoglutarate, which is an intermediate, and it yields glutamate. And then glutamate is going to form oxaloacetate, which then converts into aspartate. Okay. This is our last, yes.
So, does this include the reaction in the diagram?
Just for your question, I think I know it. So like the actual problems that you guys have, just like use the citric acid cycle and kind of do it like how I explained it to you, like through steps, how like it's a citrate synthesis, and then it goes through alpha-ketoglutarate, and then it keeps going through oxaloacetate and yields to that. Does that make sense?
So do we draw it out?
I think so. Yeah. I don't think you have to draw like all the details here, like FADH2 FAD. Just draw like this, like NAD, NADH, because just in case, because it yields to aspartate from the malate reaction. But yeah, if you have any like detailed questions, you can email me and I can like look over it. But I think that's it. Okay. Okay. Do you guys want